And finally, here, the last part of our lecture, modulation of signaling pathways. Let's think about specificity. All of these pathways have to be specific. So a signaling molecule has to bind to a specific receptor. There are many, many different types of receptors. As we mentioned earlier, the receptor determines the outcome. And every signaling molecule has its specific receptor that it binds to. There can be competition for binding sites. Think about the situation where you have a beta blocker. That's an important class of drugs, and there you have a molecule that will compete with norepinephrine for the same beta-2 adrenergic receptor in the heart. And so a beta blocker would be one that competes for the same binding site as the natural signaling molecule, the norepinephrine, would. And so they are trying both to get the spot and the binding site on the receptor, and they're competing with one another. So that would be a competition event. Agonist versus antagonist. Let me just show you the next slide here. So if you have an agonist, that would be something that causes a response. So here you would have the natural signaling molecule, the ligand right here, and it binds to its receptor and it activates it, and you get a response. So that's a normal kind of situation. Then there could be a molecule that acts just like the natural ligand, the natural signaling molecule that binds to that type of receptor, and so it acts just like it, and that would be an agonist. It mimics the effect of the natural ligand or signaling molecule for this type of receptor. It also leads to a response. An antagonist is basically a blocker, so it will compete with the natural ligand or an agonist for the same binding site, but it does not lead to the response, no response. So it blocks the activity here of this receptor, and that would be an antagonist. Now, modulation of signal pathway responses, depending on the number of receptors, the cell is more or less sensitive to a certain signal. So if you decrease the number of receptors, well, then you are less sensitive to a signal, and that would be desensitization. So you are less responsive to a certain kind of signaling molecule. Think of the situation where you first start eating very spicy food. It really hurts, and then you keep eating more and more spicy food, and all of a sudden you think, oh, I'm fine with that. I can eat a whole chili, one of those hot chilies. I can eat it, no problem, and it doesn't even hurt anymore. That is because you downregulated the number of receptors, and so the response is lessened because you downregulated the number of receptors. Similar to that would be desensitization. So you can bind, for example, a chemical modulator to a receptor, or you can multiple stimulation will cause the downregulation of receptors, and that makes you less sensitive to something. The opposite would be upregulation. In upregulation, you have more receptors that are on the cell surface, and now the smallest amount of signaling molecules will just cause a huge response. So that means you're more sensitive to a certain signaling molecule. One thing to point out here would be on the point here of drugs. The pharmaceutical industry is very interested in finding chemicals, molecules that either compete with binding sites with natural drugs or modulate the activity of certain receptors. So a very, very important class of drugs will be the beta blockers. That's a very large group of a very commonly used kind of drug to sort of blunt the sympathetic response on an already weak or elderly heart. You don't want grandma to get really upset about something, have a huge spike in heart rate. So a beta blocker will protect the heart from this sympathetic input. A brief summary of what was covered in this lecture, with the emphasis, of course, being on cell communication and the signal pathways. Now, so we're done with chapter 6, and this is also the last chapter that's part of your exam 1. So let me just give you some hints for how to study for exam 1. Please take a look at the 2 lectures that were posted online. Unit 1 is everything that we covered on site and the face-to-face lectures, and then the 2 lectures that were posted now, so on cell division, mitosis, meiosis, and a little bit on cancer, and then this lecture here. You took your lecture quiz today. I think it's still open, but it will close soon, and so that's sort of a preview of what to expect with the real exam. The real exam, exam 1, covers chapters 1 through 6, including this one, and please do not underestimate this chapter. It will take you a little time to get used to the information presented in this lecture. Give yourself some time. Right now, the exam is scheduled online to be taken on Tuesday, I believe. If you need more time, let me know. I can extend the time, but for right now, I have scheduled so that you take it on Tuesday. So you have, today's Friday. You should study this weekend and get prepared. It's a 100-point exam covering chapters 1 through 6. And you definitely want to, hopefully you started already reviewing chapters 1 through 5, including the cell division stuff. And then you want to add this lecture to it. So you should take a look at it as soon as possible, kind of start digesting the information in here, and take a look at the water-soluble versus lipid-soluble pathways, with the lipid-soluble ones, of course, being much easier than the water-soluble communication signals. The lecture format will be similar to the quiz that you took today, and just that it's 100 points worth of material. You will get an hour and a half to complete the exam, so it should be hopefully enough, 90 minutes. And let me know if you have any questions. Email me or post on Canvas if you have any kind of concerns.